Anyone?
Oh, of course. No, I did.
Okay.
We did FRQ.
Yeah.
She did it this time.
How about you, Ilana? Elena.
I did it.
Okay, you're waiting for what? Okay. Guys, electrons from the oxidation of glucose. Who knows what oxidation means?
Losing electrons.
How about in the terms of biology? Or in terms of this big molecule. Breakdown of glucose. By glycolysis and Krebs cycle, the electrons from these things are captured by the NAD plus to form NADH. So you must always have NAD plus available, right? So you must always recycle your NADH back into NAD plus by one of two ways. Either by fermentation, which means no O2, or by electron transport chain, which requires us to have O2. Now, anytime they italicize a couple of letters, that's because it's a gene. So MTND5 is a mitochondrial gene that encodes a subunit of NADH dehydrogenase. If it ends with ACE, it's an enzyme. It's an enzyme that will be responsible for oxidizing NADH to NAD+. So essentially, for NADH to become NAD+, you must have NADH dehydrogenase. Okay, this happens where? In the electron transport chain of the mitochondria. A mutation in this gene is associated with a rare genetic disorder, which results in the buildup of lactic acid in the body. There is this one researcher who made the hypothesis that the mutated enzyme has decreased activity, but it can still function to a certain extent as long as you give it a lot of NADH. To test the idea of this researcher, he would then treat a group of individuals who have this mutated enzyme with a vitamin whose structure is similar to NADH. He would then measure the concentration of NAD plus and lactic acid over 20 weeks. That's basically 5 months. Okay. These are the results, right? So there's going to be a peak in week, I think, what week is that? In week 5, there is a peak for the NAD plus. Okay. For lactic acid, it will just decrease over time. Okay. A. Describe the pattern of the inheritance that's most likely associated with a mutation in the MTND5 gene. What do you guys think? It's a mitochondrial gene. Who is responsible for passing on mitochondrial genes?
Mother.
The mother only. So we're going to say inheritance from the mother only. Maternal. Okay. Between brackets, maternal, mitochondrial inheritance. Just make sure I didn't miss anything. Can't miss a thing. This is your future here. Okay. Explain why individuals are not typically heterozygous with respect to mitochondrial genes. Well, they only get their gene from the mother, right? They only have one copy of the allele. So they only receive one allele copy, which is from the mother. And of course, if you only get one allele copy, you better be not heterozygous, right? B. Identify a dependent variable measured in the researcher's experiment. Who knows what dependent variable means by now?
It can be changed.
We're observing it. We're not controlling it. Are we controlling the time? Yes. The researcher chose to test them over 20 weeks. He could have done it in 1 week if he wanted to. So time is the independent variable. But the researcher is measuring NAD plus concentration and lactic acid concentration. So what's the answer for part B?
Concentration of lactic acid.
Concentration of lactic acid or concentration of NAD plus. Identify one control that the researcher could use to improve the validity of the experiment. So what he did was he treated this group of the disorder with a vitamin. How about taking a different group of individuals without the disorder and giving them the same exact vitamin? Or how about taking individuals with a disorder and not giving them any vitamin? That'd be good, right? That's your control. So 1 time with vitamin, 1 time without vitamin. Okay? Are we good? So basically, measure the concentration of lactic acid, NAD plus, in a group of individuals affected with the disorder but not given the vitamin. Very good.
Lisa, can you go back?
Easy. What do you guys think? Super reactive. Justify the researcher analyzing blood samples at many intermediate time points instead of at only the beginning and the end. Could you get a good study if you just analyze the beginning and the end?
No.
Right now, if I just look at you in the beginning of our course and look at you at the end of the course, would I get a clear picture of you as a student?
No.
No, because the first lesson you guys were bored, last lesson you were still bored. We have to see points of happiness, which I'm still trying to figure out where that is. Still studying you, still. Okay? To provide a more accurate description, okay, of the levels of NAD plus and lactic acid. Okay. Concentration, yeah, levels, okay. C. Describe the relationship between the concentration of NAD plus in the blood and the concentration of the lactic acid in the blood during the first 5 weeks of treatment with the vitamin. Okay. So, I think we can see clearly that the NAD plus increased in the first 5 weeks, but the lactic acid decreased in the first 5 weeks. Would you guys agree with me?
Yeah.
So basically, the NAD plus increased in the first 5 weeks, while lactic acid decreased in the first 5 weeks. Okay, based on figure 2, calculate the average rate of change in the blood NAD plus from week 5 to week 17. Okay, so week 5 to week 17. Okay, 5, 17. On the y-axis, the 5 would correspond to almost 300, and the 17 would correspond to 50, 60 or 55?
I did 60.
60, I think, is better. You're right. So basically, we're going to find the slope, just like in math. So the x-axis in the bottom, y-axis on top. So 60 minus, shall we say 290? So minus 230 over 12, which is how much? Let's see how much that is. Negative 19. Negative 19 mu mole per liter per week. Yeah. Very good? Perfect. Part D. The researcher performed a follow-up experiment to measure the rate of oxygen consumption by muscle and brain cells. Predict the effect of the mutation of this gene on the rate of O2 consumption. Guys, this gene was responsible for what? For making NADH dehydrogenase, whose job was to take the electrons out of the NADH. But now if this gene is mutated, our enzyme is not going to work so well. Will it take as many electrons as before? No. So if there are less electrons being transferred, you will also need less O2. So less O2 is consumed. Justify your prediction with less functioning NADH dehydrogenase, less NADH molecules lose their electrons. So there are less electrons in the electron transport chain, so less O2 to accept them. Because what's the job of O2? It's the final electron acceptor. The researcher had hypothesized that the addition of the vitamin that is similar in structure to NADH would increase the activity of the mutated NADH enzyme. Explain how the vitamin most likely increased the activity of the enzyme. So the vitamin has similar shape to the enzyme, so it's going to be able to bind to the active site of the enzyme, correct? So we're going to say that the vitamin has a similar shape to the enzyme, to the NADH, so it can bind to the active site. Yeah. Of the enzyme. Easy. They gave you all that to write on. That's a lot. Okay.
Yeah.
There's a disease that results when certain cells and proteins of the body damage blood vessels. Researchers continue to look for improved ways to treat people with this disease. It's called atherosclerosis. It might be possible to reduce damage to blood vessels by increasing the amount of FXR1, which is an RNA binding protein. Okay. And there's another protein called IL-19 protein that's made by certain white blood cells, which might regulate the expression of the FXR1 gene. To investigate the regulation of the FXR1 gene by IL-19, a researcher added IL-19 to certain muscle cells in the lab. And they measured how much FXR1 is there over 48 hours, or basically just 2 days. The researcher then calculated the amount of the FXR1 present at each time point compared to time 0. So at time 0 we had this much, and other times we had different stuff. Okay. A. Describe how the amino acids are categorized by their chemical properties. How are they classified by the chemical properties?
Hydrophobic.
Based on nature of the R group. So can be hydrophilic, hydrophobic. What else?
Ionic. And it's acidic or this.
Acidic, basic, or neutral. Explain how a change in the amino acid sequence of the FXR1 protein could decrease the ability of the protein to bind to RNA. Guys, the ability of the FXR1 to bind to DNA depends on the amino acid sequence of the binding site. And it has to be very specific to ensure strong interactions. If the sequence changes even slightly, the binding could end because the interactions will no longer be strong. Okay? So the binding site depends on a specific amino acid sequence to ensure strong interactions. So a change in this sequence will disrupt these interactions. B. Using the template in the space provided, construct a labeled graph that represents the data shown in table 1. Guys, I want you first of all to take some time and to do the plus and the minus using the calculator. Shall we? So do it individually. Then we're going to compare our results. .91. For now. Okay, are we looking? Okay. Thank you for the help, guys. Let's move on to the template. So, what goes on our x-axis? What goes on our x-axis? Oh, wrong.
Why?
You must have used a line graph
Why?
because we have time on the x-axis. It's not 0, but it's not full. I think you will get 2 out of 3
Why not what?
because we have time on the x-axis for one treatment.
That makes a difference?
It makes a line graph. It's okay.
That's fine. You'll not get that. You'll get it.
Time in the presence of IL 19 in hours. Relative amount of FX
Let's stop.
R1 protein. Now, my time will range from 0 to 48. I'm not going to start 0 here because it's going to be hard for me to actually draw the error bars on the y-axis because I have error bars for all the data. So I'm going to start 0 from here, which is okay. Now I'm going to treat each tick as a 2. So each 2 ticks as a 4. 8, 12, 16, 20, 24, 28, 32, 36, 40, 44, 48. Now, how about the y-axis? I must use what we call a hitch mark or a double tick because my least data is at 0.91. Use a double tick and start at 0.8. And I'm going to be making each line as a 0.2. So 1, 1.2, 1.4, 1.6, 1.8, 2, 2.4, 2.6, 2.8, 3, 3.4, 3.8, and 4. Perfect. Now, when I have 0, I have 0. Guys, look at the data. How about I send this to you so you can actually catch up with me because you guys are like, I think, a bit lost. Are you guys a bit lost?
Yes.
Oh, a bit? Okay.
Is he okay?
At least just a bit. So, 0.98. So 0 and 0.98, which is here. And I have to go as low as 0.91 and as high as 1.05. And you must now draw a line between them. Are you guys using your phones for the right purpose?
Look at me.
Come on, guys. Okay, now 4 and 1.7. 1.48. 1.92. 1.48. Yep. You see, the upper error bar has to be the same length as the lower error bar. Otherwise, they're going to minus you, so be careful. 8 and 1.6. 1.325 is my lower point, which is here. 1.875, which is here. Here. Perfect. 16 and 3.10. Are you guys good? Okay. 2.3 and 3.9. Okay. Now you see, I think it's good enough, yeah? Yeah, I think I'm doing good. Should I take the exam for you guys? Come on, man. Wake up. Okay. Now, I don't like that. I feel like the upper one is a bit longer.
Mr. They'll not bring a roller يعني.
You're right.
عادي.
Okay. 24 and 2.15, which is here. And then we're going to have lower 1.745, which is here. And upper 2.555, which is here. So here. Again, I don't like that much. Do you guys like it?
Yes.
2.15. Let's make it a bit higher here. Because my lower error bar has to be as high as the upper one. Or as long as the upper one. Right now it's a bit different. That's why. Slight difference is okay. Okay. What else?
What is gonna be?
We have 48,
Yeah.
1.6,
Plus or minus. What's your number? 1%?
1.08, and 2.12. And now we have one more thing to do, which is connect them by a straight line.
That's really big mistake. Don't do it again.
Where is it?
Very good. Okay. Now the question says,
I just found that.
do we have
Only this.
a difference in the amount of the FXR1 protein between after 16 and 24 hours? So guys, 16 hours and 24 hours. Are they overlapping? Do you guys know what that is?
Error.
No? Yes? Guys, you should draw a line between the two highlighted ones. And if that line, so look, draw a line from the lowest point of one of them to the other point. Are they hitting each other?
Yeah.
Yes.
That's called overlap. If they're overlapping, they are not different from each other. Okay? So if they're overlapping, which they are, they're not different from each other. So we're going to say no.
This is not like that.
Statistical difference.
True, it's true. You're so
Okay. Okay. Part C. Based on the data for the 48-hour period, describe the effect of the IL-19 on the FXR1 gene expression. Did the expression become more? What do you guys think?
Yes.
It became more, but not that much. Right? So it started here,
Can you help?
ended here. So we're going to say that it increased. It resulted in an increase. It did increase, right? Okay. Okay. D. The researcher hypothesizes that the FXR1 gene codes for a protein that binds to mRNAs that encodes some of the RNAs that damage the arteries. Let me explain to you what's happening here, guys, since you're like enjoying this. Look, FXR1 gene makes a protein. The protein binds to mRNA. mRNAs encode some proteins that damage arteries. So artery damaging proteins. Individuals with a particular mutation of the FXR1 gene tend to have high levels of these proteins. So essentially, when this gene is mutated and it's not working, those artery damaging proteins level up or increase. Do you know what that means? That means that the FXR1 protein was actually important. It was able to inhibit mRNA, and now without it, the mRNA has increased, or at least it's not inhibited, and now the mRNA is making artery damaging proteins. So the FXR1 protein was actually inhibiting the translation of the mRNA. Shall I repeat? Okay. So guys, when a mutation happened to the FXR1 gene, it can no longer make FXR1 protein. And now those proteins will go up. Those artery damaging proteins will now go up. You know what that means? So if the FXR1 protein is not working, the mRNA is now going to result in more artery damaging proteins. Meaning that the FXR1 protein had one job, which was to inhibit the translation of the mRNA. Which is now when it's gone, mRNA becomes translated a lot. So now we're going to say that the FXR1 protein had an inhibitory job or function, let's say, to inhibit mRNA translation. Okay. Good? So, researchers grew populations of bacteria in a growth medium that contained a low concentration of glucose and a high concentration of citrate. But citrate is not used by the bacteria. For thousands of generations, the bacteria only used glucose. Okay? And when there is low glucose concentration, they also grew to a low density. After about 30,000 generations, one population emerged that began to rapidly grow to a much larger density. The researchers hypothesized that the bacteria evolved the ability to use citrate. And if they did use citrate, they call them Cit+. To test the hypothesis, the researchers grow two populations of bacteria, a Cit+ and a Cit- in the same growth medium, which only had citrate. So what do you guys think? Which one is going to be able to use the citrate? Cit+ or Cit-? Exactly. What's going to happen to the Cit-? They will die. Because there's no glucose, only citrate, and they can't use it. They're going to die. So A, Describe one outcome that would demonstrate that given population has evolved.
Change in frequency of the allele.
Yes, change in allele frequency. B. Identify the dependent variable. What are they measuring? What are they measuring? What are we trying to observe? Of what?
The bacteria.
Exactly. Or the number of the bacteria, right? Number of the bacteria. C. Predict the results obtained by the researchers when they grew the CIT plus and the CIT minus bacteria in the medium that contained only citrate. Which one is going to grow? Which one is going to die?
We just CIT plus will grow. CIT minus will die.
Will grow. While CIT minus will die. D. The researchers claim that the CIT plus mutation increases the fitness of the bacteria. Provide reasoning to support that claim. Does it? What are you looking at?
The time.
The time? Come on, man. It makes it longer. Okay. They had to go through this like at least 3 times by now. So you should be happy. It's your first time. So can someone help us with the reasoning? How would having this mutation increase the fitness of the bacteria? What? Okay. Oh my God. They are able to consume a food source that's not available to other bacteria. So this means less competition, more chance to survive and reproduce. Question 4. The Tasmanian devil is a species of mammal whose only wild population is native to the isolated island of Tasmania, off the coast of Australia. Tasmanian devils have a very low level of genetic diversity. Is that a good thing?
Low level of genetic diversity.
It's horrible. It's horrible. Researchers hypothesized that this lack of diversity is because of genetic bottleneck. Can someone give us an example of genetic bottleneck?
A fire?
Yeah, like a fire that killed many of them, or a virus, or hunting, or some other disaster. So in 1996, there was a disease called the devil facial tumor disease, which appeared in this population of Tasmanian devils. This virus was able to, or disease, was able to spread between them and cause the death of most affected animals. The impact of the disease resulted in a decrease in population of more than 80% of them. Okay? About 90% basically. Over 20 years. A. Describe the process that maintained a stable population of these organisms before the appearance of the tumor in 1996. What are the things responsible for maintaining a stable population size?
Carrying capacity.
Can you elaborate?
I guess.
Can someone...
Carrying capacity.
Can-
Sexual reproduction.
Can someone elaborate on those claims? Okay, elaborate on your claim.
I just said anything. I don't know.
How about saying density dependent factors that made birth rate equal death rate? How about that? B, explain how the huge reduction of this devil population since 1996 affects the ability of them to get new diseases in comparison with population before 1996. What happened in 1996? There was a new disease that killed a lot of them and reduced their genetic diversity.
Yes.
Did this disease make them more prone to infections? To other diseases? Look, if you have 10,000 individuals that become then only 1,000, which side has more chance of getting a disease?
1,000.
Why? Because we have less genetic diversity, so more chance of getting sick.
Musa, can you repeat?
Yes. Whenever you have less numbers of organisms, that's a bad thing. Because that also means we have less diversity, which means more chance of getting sick.
Done.
Yeah. So this guy has a big problem, the 1,000, I mean. So we're gonna say with less genetic diversity, there is more chance of getting new diseases. How do you guys like the handwriting? Okay, let me just work on look at the answer key, make sure we're not making mistakes. Can't afford to make mistakes.
Yes, ma'am.
Okay. C. Tasmanian devils are top predators. So you know what that means? They're top of the food chain. And they're considered to be a keystone species in their community. You guys know what that is? A keystone species?
Like they depend on another species.
No. The ecosystem depends on their existence, even in minor numbers. Without them, the entire ecosystem is gonna fail. Predict the effect of the rapid reduction of this population on the rest of the community. Will the community benefit or get harmed?
Harmed, of course.
Yeah. So the community will collapse. D. Justify the prediction. What's the reason that the ecosystem will collapse?
Because they depend on them. And
Who's they? Never say they in the exam, by the way, without saying who's they.
-1.
Can you elaborate? Do we have one student here? How about the rest? Who's grade 9 here? Who's grade 10?
Error.
You're not grade 10?
I'm good.
Okay. Can you please help us? Where's Farah? Her mother is cooking something, that's why she didn't come.
And was that what she said? No way.
Mr. She told me outside, Mr. Wesley, my mom's cooking. I'll not come.
Cooking for the guests, yeah. She's gonna hear this,
I don't know.
I think. Okay, moving on. Her mother is cooking, not her. Okay. So, look, it seems like these devils, can you actually Google them?
Yes.
Google Tasmanian devils and see what they look like. You will like the picture, I think. Let's see. Wait, does this thing work? How do I talk to my device?
Oh my god, this is I love the
Hey, Bixby.
It's a dog?
What's the picture of a Tasmanian devil?
It's recording.
Okay, here's what I found online.
Do you guys like the picture?
Yeah.
Whoa, that doesn't look like a dog.
No,
Whoa.
My dog's pretty.
Look, that's just evil looking. I mean, look at those teeth. That's not. They look like devils. Look at those ears. Ooh. Okay. Rocky. Who's that?
He said that's my dog and she's making fun of it.
You named your dog Rocky? Really? Okay. The Tasmanian devils will no longer feed on the prey species that will now become over abundant and will overeat their own food. Look, is having a lion in a forest important? Why not just kill all the lions? Because they kill other animals. If you kill all the lions, you're gonna have way too many herbivorous animals. And these guys will just eat all the available vegetation and they're gonna go through extinction because they will finish all their food. You must have something that will eat them to limit their numbers. So you're gonna always have vegetation. You must always have a keystone species. Okay. There's a protein called P53 protein, which is a transcription factor that regulates the cell's progression through the cell cycle after DNA is damaged by UV light. Okay. So P53 will bind to the DNA. That's why it's called a transcription factor. And it will result in the production of cyclin-dependent kinase CDK inhibitor. Who knows what this inhibitor does? What's his job? To have more cell division or to have less cell division? If it's inhibitor of cyclin, less cell division. So P53 is important ultimately to cause less cell division. It's important to stop cancer because cancer wants to have lots of cell division and this will simply stop cancer. A skin cell completes one round of the cell cycle. Describe the products. What are the products of mitosis?
Diploid cells.
2 genetically identical diploid cells. B. Based on figure 1, explain how P53 regulates the cell cycle in the presence of damaged DNA. Guys, if there's damaged DNA, P53 will make CDK inhibitor. Why? To stop the cell cycle. So it will cause the expression of the gene that codes for CDK inhibitor to stop the cell cycle. C. Draw an X on the template in the space provided for a response to indicate the phase during which the replication of damaged DNA would occur. Guys, DNA replication happens in which phase?
S.
S. So put an X on this. D. Based on figure 1, explain how a mutation to P53 may lead to an increased risk of cancer. Well, P53 stops cancer by making CDK inhibitor. But now, if it's mutated, can it stop cancer?
No.
No, because it can no longer make CDK inhibitor. So it can't bind to DNA, so it can't initiate the expression of the gene expression that would make CDK inhibitor, so it can't stop cell cycle. That is D.
Mister, can you re-explain B?
Yes. Explain how P53 regulates the cell cycle in the presence of damaged DNA. Look, when DNA is damaged, what happens? A protein kinase is made. The protein kinase makes the P53 go to the nucleus, bind on the DNA, which will result in the making of CDK inhibitor, which means inhibiting the cell cycle. So if DNA was damaged, your body will try to avoid cancer by stopping that damaged DNA of that cell from dividing in the first place. And the cell will not divide, it's not going to become cancer. So your body tries to protect you from having cancer. Okay. Take a minute to read this. It is not the easiest thing in the world. Okay. The human kidney filters the blood to retain, which means to keep, useful substances and to remove waste products along with excess water as urine. We have two genes, SLC3A1 and SLCA79. They're two genes that encode one part of a transporter protein that's typically found in the plasma membrane of a human kidney cell. This transporter complex moves certain amino acids across the plasma membrane. And if those two genes are mutated, this will result in more polar amino acid cysteine in the urine. So that means that those two genes are responsible for removing this amino acid out of urine. So if they are mutated, this amino acid will stay in the urine. Okay. We are discussing 7 individuals who have a family history of this disease, which is called Cystinuria. The researchers measured the concentration of cysteine in the urine of the 7 individuals. And they also found the sequence of the genes to know if they have functional alleles or not functional alleles. Now, individual 1 has the most levels of cysteine in the in the urine. This individual is very damaged. This individual has 2 non-functional alleles of the SLC3A1, has 1 good copy of the SLCA9 allele. So individual 1 is the most damaged individual. So the answer to A is going to be individual 1. B. Describe the relationship between the total number of mutant alleles in individual and the concentration of cysteine in the urine. Can someone tell me which individual here is the most affected?
One.
Who has 3 negative copies. How about individual 2? The second most, who also has 3 negative copies. But then as you have 2 negative copies, the number drops. If you have 1 negative copy, the number drops even further. Correct? Okay. So basically, if we have more negative copies, which means mutant alleles, we're going to have more cysteine concentration. So we're going to say more mutant alleles leads to more cysteine concentration in the urine. Evaluate the hypothesis. Evaluate means it could be wrong or it could be right. Evaluate the hypothesis that mutations in the SLC-A9 have a greater impact on the transport of cysteine across the plasma membrane than the mutations of the SLC-A1. Do the mutations in the SLC-A9, are they worse?
No.
How do you know?
Because like the first individual has 3 negatives and the second one as well, but it's more affected in the first one.
Smart. So if you guys compare individuals 1 to 2, individual 1 is mostly damaged due to having 2 negative copies of the SLC3A1 allele. Individual 2 is damaged but not as damaged for having 2 negative copies. Okay? So we're going to say not supported because individual 1 with 2 mutant SLC3A1 has more cysteine than individual 2 with 2 mutant SLC7A9. SLC7A9 alleles. She's right. Right? So having 2 copies of SLC3A1 is more damaging than having 2 negative copies of SLC7A9. Get it? No, yes? Okay. So look. I think we can all agree that individual 1 here is the most affected. And we can tell that having 2 negative copies, which means having 2 mutated copies of the SLC3A1 is worse than having 2 negative copies of the SLCA79. 32.2 is worse than 29.8. What causes 32.2? Having 2 negative copies of A1 and having 1 negative copy of A9. What causes less damage? Having 2 negative copies of A9 and 1 negative copy of A1. So in conclusion, having 2 negative copies of A1 is worse than having 2 negative copies of A9. They're close, but it's worse for individual 1. And the hypothesis says that having negative copies of A9 is worse than negative copies of A1. We just showed them that having negative copies of A1 is actually worse. So we're actually going against the hypothesis. We're going against it. D. Explain how the data support the claim that cysteine is a large polar molecule. Guys, if we have a large polar molecule, does it need help getting transported? It does. It needs the help of a transport protein. Who is making the transport protein? A1 and A9 genes. So yes, when having or just having a mutant copies of SLC7A9 and SLC3A1 alleles leads to buildup of cysteine in the urine due to lack of transporter protein. Needed to move cysteine out of the urine.